So I will talk about metabolic individuality across scales, from single cells to microbial populations. And I'm currently a postdoctoral researcher at JAMSTEC, but I was a student at ELSI, and big portion of my talk is about work that I did at ELSI together with Sean. So to set the stage for my research and maybe also put my research into context of the symposium and talks that we've heard earlier this week, I would like to talk about individuality in the context of ecology for a bit. So in ecosystems on Earth, you can choose your favorite one. I'm a microbial marine ecologist, so I prefer marine environments, but you can choose terrestrial one. We have different groups of organisms. So we have primary producers such as algae and plants. We have consumers that consume those plants, so zooplankton or small animals. We heard about bees earlier this week as well. And we have carnivorous consumers like small fish and smaller land animals. And then we have the top predators at the top of the food chain. And they're all individuals as part of the ecosystem. We can see each group as an individual part of the ecosystem. We can see individual species within each group as part of the ecosystem. And within each of these species, we can then see the individuals as part of the herd or population. So for example, for fish, we can see an individual fish as a part of a school of fish. Or for bigger land animals, we can see individuals as part of a herd. And their behavior, individual behavior of course is important, but their behavior also is important to sustain their community. So when Sean asked me to talk about my research at the symposium, I was trying to see where did I look at individuality in my past research. And just a few examples of something that I did at ELSI, for example, was I looked at genomes, so individuality in genomes. This is for methanogenic archaea, and here we did genome comparisons. So looking at 86 species of methanogens and comparing their genomes, how similar or dissimilar are they? So here you can see each bar represents a species, and on the y-axis you have number of orthogroups, so number of genes with shared ancestry and shared function. And the blue part is what all of these species share. The yellow part is what some of them share, but not all of them share. And then the green part is what makes each species individual. And when we compare the species at different phylogenetic levels, so seeing how much of their genomic content is shared pairwise within each of these phylogenetic levels, we see a decrease in similarity with increasing phylogenetic distance. So this is just one example of individuality in the context of genomes for methanogens. And then also for methanogens, I looked at individuality in their metabolism. So I did stable isotope probing and looked at how individuals differentially take up carbon and nitrogen in their metabolism. So here you see a picture of 59/49 ratios. This picture was taken with the nanosims in an aggregate of methanogenic archaea. And you can see that cells on the outside tend to take up larger amounts of the substrate than cells on the inside. So there seems to be some shared, some division of labor, which we also heard about earlier this week. Then what I'm doing now is a bit different. So one of the projects I'm currently working on is genetic recombination. And all of the experimentalists among you, especially biologists, will know that a lot of the stuff that's happening in the lab, a lot of times it's kind of luck or chance, and things sometimes happen without us really understanding why they're happening, especially in work like isolation. Sometimes you're lucky and you get an isolate of a culture of a microbe. Sometimes you're unlucky and you don't get it. So there's some luck. And with my genetic recombination work, so the cultures that I work with, this is kind of the default color. But then when I do the genetic recombination, some of them turn bright red and some of them turn green. So there's some phenotypic variation that is kind of hard to understand because genetically these should all be the same. So this is just the pigment change. And then another example for phenotypes is cyanobacteria, and these are cyanobacteria that I'm currently working with. I did TEM imaging on them to show the internal arrangements. So this is a model marine cyanobacterium. You can see the internal structure. This is the thylakoid membrane. so where photosynthesis happens, then you have some internal structures, DNA and polyphosphate granules. And these are pictures from the same strain, same sample, just different cells. And you can see the internal structure is different between them. So sometimes the thylakoid membrane is continuous and sometimes it's thicker. And then when we look at dividing cells, also their internal arrangement is a bit different. So there's a lot of variation internally as well in the cells that we don't see just looking at them with the normal light microscope. And a final example is the work I'm currently most focusing on, which is metabolic individuality in cyanobacteria. So I'm comparing different species of cyanobacteria, and I'm trying to get their metabolomic fingerprint. So what kind of metabolites do they release, and how does that vary between species, but also within species, between growth conditions and the stages of their growth cycle. And I will talk about this later in my talk. So these are kind of the exometabolome fingerprints of the cyanobacteria species that I'm growing now. Yes, you can see I've worked with different types of organisms in the past, and what I'm currently working on and what also will be the focus of my presentation is cyanobacteria. So this is a genus level tree of all of the cyanobacteria from GTDB, and the marked branches here are the species that I'm working with. So I'm working with ancestral species, freshwater species, I'm working with marine species, and they have very different morphologies. So some of them are single cells, some of them form aggregates, some of them are filamentous, so they form these long chains, and they might have heterocysts in between, so maybe differentiated cells. And then some of them are the filaments, and they have very big polymer layer around them that keeps the filaments together. For the first part of my talk, I will focus on this species, Synechocystis. It's the model freshwater cyanobacterium. And then for the second part of my talk, I will focus on comparing these species of cyanobacteria. So to bring us back to this slide, for the first part, I will look at individuals as part of their herder population. So cyanobacteria, so it will be individuals as part of their population. They don't form herds. And then for the second part, I will look at the species within each group. So looking at primary production in the ocean, how do cyanobacteria differ in their exometabolomes. To get us started on the freshwater model cyanobacterium and individuality in the population, I would like to explain the concept of phenotypic heterogeneity, which is that individual cells, even in a genetically homogeneous population, have different phenotypes. And this is not a direct response to environmental change. The heterogeneity may increase or decrease with environmental conditions, but it's kind of the cells betting on something to happen and exhibiting different phenotypic states so that if environmental conditions suddenly change, the population can survive. So this is beneficial for population survival. Possible causes are stochastic gene expression, asymmetric cell division or cellular interactions, and the effects are increase in population growth, division of labor, and functional diversity and survival. So a lot of the studies in the past have done extensive work on observation and quantification, which is more or less straightforward. It's difficult at the single cell level, but people have done it. But there's a lot of uncertainty about these underlying causes. We don't really understand what's going on. So in my master's, what Sean and I were interested in is to kind of see if we can find explanation for the heterogeneity at the metabolic level. So what I did for that is, as I told you, I worked with this model freshwater cyanobacterium Synechocystis, and I did cultivation, high-resolution mass spectrometry to get to the map that I showed you before, and then integrated that into a metabolic model. And the work I will talk about in the next maybe 15 minutes is based on this preprint that is available and that we're currently revising together with a group in Europe that we will later have a talk by Kepa, who is also from the same group as Ariane and Daniele. So to explain the concept of the preprint, basically we established a new pipeline, and it's a very long pipeline, so I will walk you through it step by step. We started with the experimental part. So I grew freshwater cyanobacterium in standard medium vials and stressed medium vials, so lowering nitrogen, sulfur, oxygen. Gas inflow was also different. And then I added isotope label, so heavy carbon, nitrogen, and water to trace the metabolism of these organisms. I then did nanosims analyses to get, like the picture that I showed you before, maps of individual cells, and we can count the ions of both the heavy and the light isotopes on individual cells, and we can get the ratio of heavy to light isotope for each cell to approximate how much of a compound they take up. So to put this in kind of the global context of our project, cultivation and bulk growth analysis, and then looking at the single cell isotope incorporation, knowing the final and initial isotope ratios as well as the natural abundance, we can then calculate the uptake of isotope of substrate into each cell. And we can plot that and we can get a distribution that looks like this. So we have both the standard and the stressed condition. The red line is if C and N uptake would be 1-to-1, so if cells would take up equal amounts of C and N, which we see most of them do not. So we see, first of all, differences between standard and stressed cells, a lot less uptake of carbon in the stressed cells. And we also see within each of these groups, so within the standard and the stressed condition, cells do not behave the same. So we have this big cluster of cells here, but we also have some outliers here. And the first thing we did to look at this data is what previous studies have also done. We looked at the variation, so the phenotypic heterogeneity, quantifying it, and looking at the variation in uptake rates between the conditions and also between carbon and nitrogen. And then we also calculated the coefficient of dispersion, which is kind of like a measure of the heterogeneity, how spread out are the data. And we can see that, especially in the stress condition, there's high level of heterogeneity in the nitrogen, but overall also heterogeneity in carbon and nitrogen in both of the growth conditions. So to take this a step further, this is also something that previous studies have done. We tried to get to the growth rate. So we have carbon and nitrogen uptake data into individual cells, and from that we can approximate the growth rate. Previous studies have done this using only the isotopic ratios, so only looking at the isotopic ratios, but because we wanted to have both the carbon nitrogen data incorporated into the growth rate. This is when we reached out to the group in Europe and asked them to help us use their approach, which is maximum entropy, to calculate single cell growth rates. So how does this work? We have the cell and we have the uptake rates of carbon and nitrogen, and we assume that a lot of it will go into biomass, so growth. And we can also assume that some gamma of it will be turnover. It can leave the cell, it can go somewhere else in the cell. It's not biomass. And we assume that the maximum growth rate of each cell is limited by the minimum uptake rate of carbon or nitrogen. So to grow, a cell needs a certain amount of carbon and nitrogen, and the lower one of them in each cell is the limiting factor for the growth rate. This picture, Liebig's law of the minimum, you might remember earlier this week we had a talk from Takeyama-san about global constraint principles for microbial growth laws. So here this more elaborate figure of the law of the minimum. We are using bit simplified version, and we calculated the growth rates and get distribution like this. So for standard growth, unimodal distribution, and for the stressful growth, we get kind of bimodal distribution. So now we have 3 parameters for each cell: the growth rate, carbon uptake, and nitrogen uptake. And we did a lot of work to get this for individual cells, but now we're going to cluster them back together into groups because it's hard to understand concepts for hundreds of individual cells. So we try to cluster them into groups that have similar metabolic activity. So here you can see a 3D plot of the growth rate and nitrogen and carbon uptakes, and each cell plotted into it. The polytope is a metabolic model boundary imposed onto these cells. So theoretically, using a metabolic model, which space could the cells occupy? And you can see that the cells kind of cluster. So we have 4 different clusters of cells. We called them cone and I think noche. And they are basically nitrogen optimal, carbon excess, or carbon optimal nitrogen excess. So what that means is, if you look at these 2D simplifications of the left plot, you have a line of cells here. These are the nitrogen optimal carbon excess ones. And they basically have the lowest possible nitrogen uptake that they require for growth. So they follow this line, and if they take up less nitrogen, they will not survive. But they take up more carbon than they needed. So in this plot, this is carbon plotted against the growth rate. They take up more carbon than they would need. If they would optimize for both carbon and nitrogen, in both of these plots, they would be along this line. And then on the other hand, we have the cone clusters, which optimize for carbon. So they take up as little carbon as possible, but more nitrogen than they would need. And for each of them, we also have few cells that follow similar metabolic structures, but have very low growth. So we have these 4 clusters, and we're interested to see what exactly is happening in each one of them. To do that, we apply a genome-scale metabolic model. We heard earlier this week also from Rebecca about metabolic networks. So I will spend a few minutes to explain to everyone how we can use our data to incorporate it into the model and make the flux balance analyses more accurate. So what is a metabolic model? This is human metabolic pathway map from a paper that was published 7 years ago. You have different metabolic subsystems. They're all connected. And in a genome scale metabolic model, it's basically a mathematical representation of an organism's metabolism. So you link metabolites to each other by reactions, and there's also a matrix underlying this that links the corresponding enzymes and genes. And to make the experiment that we did a bit more touchable, to make it easier to understand, I always like to use this analogy. So this is the JR East railway network for Greater Tokyo area. I believe we are somewhere here. And we can make a small thought experiment. So let's see, we want to know how much of a certain metabolite is produced. So let's say we want to know how many people can commute from Kanagawa to Tokyo between 7:30 and 8:30 a.m., which is peak hour. And we can look just at the Keihin Tohoku line from Yokohama Station to Shinagawa Station, which is the blue line going here. And we can get the flux by standing at the platform of Shinagawa Station and counting all of the people that come off the train. So for 1 hour, we stand at Shinagawa Station, Keihin Tohoku line platform, and we count all of the people that come off the train. And let's say there's 20 trains arriving, and a total of 50,000 people come off, assuming all trains run at 100% capacity. Now, what if we change the number of trains? So we can change the flux boundary, we can change the input into the system for Shinagawa Station, for that metabolite that we're interested in. There might be some delay on the train line, so then less trains run. Or there might be some major events, so JR increases the number of trains. So if we have only 10 trains, 25,000 people can come to Shinagawa Station. But if we have more trains, we can count more people at the station. Now, in my experiment, I told you that I use stable isotopes to track metabolites in the network. And basically what you can imagine this as it is, at Yokohama Station, you give a certain number of people bright yellow helmets. And they wear these helmets on the train, and they get to Shinagawa Station, and then you can see how many people with bright yellow helmets get off at Shinagawa Station. And then maybe you have more people that can count at different stations in the network. So maybe some of them will transfer to the Yamanote line. And then you can put someone else at Shibuya or Shinjuku Ikebukuro station. And you can count how many people with yellow hats get off at each of the station. So in that way, you can track the people in the system. And in a similar way, we use stable isotopes to track the metabolites in our metabolic network. And we can adjust the boundaries of individual reactions using our data of knowing how much carbon or nitrogen do the cells take up. These networks are very sensitive to flux changes, even in a single reaction. So if we double the amount of people going to Shinagawa, they have to go somewhere. They have to either go to work here, so they will leave the train network. the cell in our case, or they will continue on to different stations, different reactions. They will be transformed into different metabolites. And we incorporate our experimental data to make our metabolic model for freshwater cyanobacteria more accurate. So we do that, and in our first attempt, we just run the model, assuming each single cell is an individual. They are not connected. But if you remember the 3D plot, there are some cells that are outside of the theoretical boundary. So in theory, they are not supposed to be alive because the model tells us they shouldn't be. So what is happening here? We thought that if the cells produce these acids, they must go somewhere. If they just continuously release the acids into the media will be acidified and not suitable for microbial growth anymore. So after considering this and that there are some cells out of the boundary, we allowed for exchange between cells. So rather than seeing each cell as an individual and running the flux balance analyses, we now allowed for cells in the network to interact with one another. So they can exchange material, they can release into the environment, and they can also take back up from the environment. So in our case, we are considering the exchange of organic acids. Assuming the cells take up CO2, they release it as organic acids into the environment, and then some other cells might take it up and use it for their metabolism. Re-release it or just keep it. So if you remember the gray cell from the beginning, now we have bidirectionality here. So there's a carbon flux different from just CO2 or bicarbonate from the medium. There's a carbon flux of organics that can be taken up from other cells. So there can be exchange between the cells. And if we do that, if we allow for that exchange and rerun the model, we see that some cells, they take up, so negative acid flux means release and positive means uptake. So few of them release a lot and then others take up what they release. And considering my culture density, we modeled this into a 3D space and looked at the interactions between individual cells. So now for each of these cells, we have the metabolic model solution, which is a flux balance analysis solution. So matrix of all of the fluxes through all of the reactions. It's a couple of hundreds of reactions. And we can see how their metabolism varies. So the final step in this workflow is to characterize the metabolic activity and the differences between the clusters in the interacting population. So for each of the hundreds of reactions in the model, we can do plots like this, where we have our 4 clusters, and we can plot the flux of the reaction with the growth rate. So we get this is glutamine synthetase, so nitrogen assimilation. We have data for nitrogen, so this might be a good reaction to look at. And we can do that for other reactions as well, like the Rubisco or energy generation or some stress marker production. And we can do that for hundreds of reactions. So we look at the individual reactions, but again, it's kind of hard for our brain to process information from hundreds of reactions. So we group them into metabolic subsystems to make it a bit easier. So then we get this heat map of our 4 clusters and the flux differences through these metabolic subsystems. These are now a bit more easy to understand. So we have, for example, photosynthesis, glycolysis, carbon fixation, and also some amino acid synthesis metabolisms. And we can look at the differences between the clusters and how in which metabolic subsystems they differ and compare them. And then for each of these subsystems, we can make a map like this and see, okay, which individual reaction in the subsystem is most responsible, so thick arrows, for the differences that we observe. So you have the map, and then for each reaction, you have the average for the different clusters. Okay, so to summarize this part, I think we were successful in looking at the mechanisms that cause phenotypic heterogeneity, at least the metabolic variation of these individual cells. And we established a new pipeline to integrate single-cell isotope data into genome-scale metabolic models. we observed a higher degree of heterogeneity during limited growth and also different degrees of heterogeneity between carbon and nitrogen uptake. And we also found that the exchange of material is essential for these populations and also for the metabolic model to work. So the next part of my talk, I will spend talking about my current research, which is looking more at the final part of the preprint that we have available, the exchange of material. So in my current research, I try to use metabolic models in marine ecology. And there's a very nice review from last year that was published on how the metabolic models can help different branches of marine ecology to predict future changes or to predict interactions in the system. And one that I'm particularly interested in is the release of material so which metabolites are released by cyanobacteria into their surroundings. And this paper suggests that the release is important for biogeochemistry, so for carbon cycling by material being released and sinking down. But what we are really interested in is how is this important for consumers? So there might be another path where it's not only going into the cycling of carbon sinking down like physical cycling, but there might be more biological cycling. So the release of material might be important, not only for sinking, but also for reuptake by other organisms. And to do that, I told you a bit earlier already about metabolomic fingerprinting of cyanobacteria. So my workflow is similar, but very different because the methodology I use is very different. I use cultivation of now not one strain of my cyanobacteria, but multiple strains. And I do orbitrap analyses to see, okay, they take up CO2 and water and sunlight, and they release oxygen. This is what's very important for the evolution of biogeochemistry on Earth. But they also release metabolites. So I want to get to the diversity and quantity of these metabolites. Some acids, some lipids, some biological building blocks. And then I use that data and integrate it back into the metabolic modeling. So the overall workflow is kind of similar to what I did at ELSI, but the core experimental step is very different. And then I get to this metabolomic fingerprinting of the different strains that I work with. These two are filamentous cyanobacteria, and these two are not. This is a model organism, and for each one of them, I have data for 2 different growth conditions, A and B, and then different time points also in their growth stages. And we can see that their fingerprints are very, very different. And when we try to connect those fingerprints back to the morphology, so doing dissimilarity matrix for all of the strains and their growth conditions, we get to clustering of the filamentous strains together and the non-filamentous strains together on the principal component axis 1. So there seems to be some connection between organic matter production and the morphology of strains. And I'm also trying to track this back to the evolutionary history of the clades. So going back here, some of the strains I have are very deep branching. Some of them are modern strains. So by looking at what they produce and which pathways are available at which branch in the tree, we can maybe see when did certain metabolites become important in Earth history, not only for cyanobacteria, but also for their heterotrophic consumers. Okay, so the final part of my project is the incorporation into metabolic models. So I use PCC 7002, which is model marine cyanobacterium, for this part of the talk. I'm establishing the pipeline with this one and then later applying it to all of the other strains as well. So for this metabolic model, I have my experimental setup, so media composition that I can use to constrain uptake reactions. I have my metabolomics data, so the amount and variety of compounds that they produce to constrain the release of, so exchange reaction in the other direction, release of material into the environment. And I also have transcriptomics RNA-seq data that I can use to constrain internal reactions in the network. So considering all this, I now have a lot better constrained model than originally published one. And I ran flux balance analyses for this model for the 2 different growth conditions and also for different environmental conditions. So here you can just see, similar to in the first part of my talk, a heat map of different metabolic subsystems for the default published model, and then standard medium modeling, low nitrates, so nutrient-limited growth modeling. And then also I tried to maximize CO2 uptake because that's something that's important in ecosystem conservation and global change. So I get this kind of data. Again, I get it for hundreds of reactions. And I'm hoping that as a next step, I can feed this data into environmental scale models. So we're working together with other people in JAMSTEC and Tohoku University that will hopefully in the future be able to use my data to integrate it into ecosystem scale, environmental scale modeling, not just the metabolism of individual strains. Okay, so I hope I convinced you that metabolic models can help us understand how populations are made up of individuals. We looked at the metabolic individuality of single cells in pure cultures through individual reactions and also division of labor in the aggregates of organisms. And we also looked at the metabolic individuality of species contribution to their surroundings, so the metabolomic fingerprints and the differences in growth conditions. So to maybe summarize all of this, it is important to look at individuals in a population, but I think it's also important to see the individuals as part of their population, because the reason for them to be individual is probably to sustain the population and to sustain the survival of the species. So of course we should look at them individually, and we now have the tools to look at individual cells, the uptake of individual cells, but we also need to contextualize that within their population, division of labor, and also in higher organisms, the social behavior of the animals. So with that, I would like to thank collaborators and also ELSI for having me again. This is Sean Lab in 2019. I think it has grown a lot since then. All of the people except Sean are now gone. So it's good to see that the lab has grown over the past years. And a lot of the work that I presented, the heavy lifting for the modeling, was done by Ariane and Daniele. Kepa, who will give a talk later, is also working with them. And then my current work is done at JAMSTEC in Imachi-san and Nobu-san's lab. And I would also like to acknowledge the funding agencies for the later part of my talk. So with that, I will leave you with this slide, and I'm happy to take questions.